human kidney. So our spatial omics studies focus on glycosylation, which is a ubiquitous protein post-translational modification. And glycosylation plays a key role in regulation of protein function, cell adhesion, and immune response. Aberrations within glycosylation have been linked to various diseases such as inflammation, autoimmune diseases, and cancer. However, underlying mechanism behind it and the structure of these glycans is not known yet. There are two main types of glycosylations that we know. One is O-linked, where the sugar or glycan is attached to serine or threonine residues of a protein, or N-linked glycans, where the sugar is attached to asparagine residues of a protein. And glycosylation plays a role in renal structure and function, and the study can provide insights into pathophysiology and clinical applications. As such, it's important, as such, imaging and glycans at the cellular level and functional tissue level resolution is essential to understand glycoprotein function. One of the most widely used techniques for glycan imaging is MALDI mass spectrometry imaging. And previously, we used cell-based features defined by Codex imaging to determine glycan composition for human lung and human lung functional tissue units. Our goal was to do something similar for more clinically common assays, such as immunohistochemistry or PAS staining, as shown here. And with emerging AI tools, we now have AI-based tools for functional tissue annotations. And today we'll be talking about a tool called ComPreps that have been developed for kidney digital pathology. So combining these functional tissue annotations with MALDI mass spectrometry imaging can help us to determine disease-related and linked glycosylation changes within specific anatomical regions and cells. And in order to achieve that, we've come up with the following workflow for spatial omics analysis of kidney tissue, where we first take our FFPE tissue section and prepare it for mass spectrometry imaging. We then perform MALDI mass spectrometry imaging to obtain spatial and glycomics, and we upload it to an open source software called Metaspace for FDR-controlled and glycan annotation. And lastly, we upload it to another software called Skills for mass spectrometry imaging data analysis. The same tissue that has been through MALDI mass spectrometry is then stained and imaged with microscope. That microscope image is then sent to our collaborators to run it through an AI-based tool for functional tissue unit annotations. Then we open those annotations and perform quality control on them in the software called QPath, where we can also convert our file into a type that's compatible with our mass spectrometry imaging software Skills, where we overlay the two pieces of data, one from mass spectrometry and the other from an AI-based tool for functional tissue annotations. And lastly, we then export it into our programming language to study the glycan upregulation and their structural analysis. To give a little bit of a background about matrix-assisted laser desorption ionization mass spectrometry imaging, or MALDI-MSI in short, for glycan spatial profiling, we first treat our tissue with an enzyme pentase F to cleave off our sugars off of a protein as shown here. And it's a multi-step process, and you can access a full protocol in the following resource. We then run our tissue at timsTOFlex mass spectrometry instrument, where our tissue gets ablated by a laser and it creates a spectrum with mass to charge ratio and intensity. And that spectrum gets uploaded to an open cloud software Metaspace for FDR controlled annotations based off of this glycan database. And these steps are completed within our lab by Dr. Dusan Velichkovic. Then we stain our tissue with PAS staining and image it using microscope. This tissue is 2 micrometers thick, and send it to our collaborators at University of Florida to a group led by Dr. Pinaki Sardar, where they've developed a tool called Computational Renal Pathology Suite, or ComPreps, which is a cloud-based open access tool with user-friendly interface, which can detect microanatomical structure in kidney tissue section and can process tissue previously analyzed by MALDI mass spectrometry imaging. And you can see here some of the results of the AI-based annotations, and you can see some co-localization between 2 images. For the purpose of our research, we've chosen 3 functional tissue units: glomeruli, tubules, and arteries. And so compreps will create JSON annotations that have to be then converted to a QPath GeoJSON format, and you can find a tool for a conversion in here. And GeoJSON format and QPath GeoJSON format are slightly different, so it's important to convert it into QPath GeoJSON format. That GeoJSON file is then dragged and dropped into QPath as shown at the picture in right. And then you can see those annotations imported within QPath. We then perform quality control. And then in QPath, we install yet another extension where you can find a full protocol in the following resource in order to export this file in a format that's compatible with our mass spectrometry imaging software skills. And so now after all those steps, we can combine two pieces of data within skills software. One is mass spectrometry imaging results with the ion map of N-glycan distribution, and compreps functional tissue annotations. And in this example, I'm showing glomeruli. And once we overlay the two pieces of data, now we know N-glycan abundance within a certain tissue functional unit. Then after we overlay two pieces of data, we export it into an R using a skills API, and we will use a package created here by PNNL by Dr. Jeremy Clare called Aromics Processor, where we first import our data and all the libraries. We then process this data into a table and normalize it using logarithmic transformation and median centering. And then we perform two-tailed students t-test in order to find upregulated N-glycans at the regions of interest. And we can then visualize all those results using box plots. Another tool that has been developed here at PNNL as well is called N-glycan Minion, which you can access at GitHub, or in full N-glycan mining and ontology, which will take all the N-glycans that are regulated within the regions of interest and then classified into the following classes using these conditions. And you can see some of the examples here. So we first tried out this workflow on a healthy reference tissue, and we were able to find the following results of these N-glycans upregulated at artery, glomeruli, and tubules. And you can see an example here. And we were then able to classify and see the N-glycan abundance per each functional tissue unit in here. We then moved on and integrated samples with disease categories such as acute kidney injury, diabetic kidney disease, and diabetes mellitus resilient samples. And we were able to find the following results. For glomeruli, these are just some of the examples of upregulated sugars. We found out an enrichment of polylactosamine, which is not present within normal glomeruli. We found an enrichment of galactose-containing N-glycans for DKD glomeruli samples, and we were also able to find the enrichment of monofucosylated N-glycans within DMR glomeruli. For arteries, we found an enrichment of sialic acid present at AKI arteries, and sialic acid has been reported in literature as responsible for as an N-glycan responsible for inflammation. We're also able to find ambiguous bisecting and tetra and ternary N-glycans only upregulated within DKD arteries, and enrichment of sialic acid also at DMR samples. In tubules, we found that polylactosamine was only enriched at AKI samples, and high mannose was enriched for DKD tubules. And lastly, sialic was enriched at DMR tubules as well. In summary, this workflow increased the throughput of analysis by integrating AI-based tools for functional tissue unit annotation and integrating multi-mass spectrometry imaging with tools such as compreps helped us to define N-glycomic composition in glomeruli, tubules, and arteries within kidney. This workflow allowed us to also define functional tissue unit heterogeneity between different disease states. And in future, this workflow can be applied to studies of other spatial omics, such as lipidomics and others. And as digital pathology tools improves, we can also apply this workflow for other organs, such as human lung. That completes my presentation, and I would like to thank my mentors, Dr. Chris Anderton, Dr. Brittany Gorman, Dr. Jeremy Clare, and entire Anderton team at Pacific Northwest National Lab. And I would like to thank HubMap for this incredible opportunity.
Wonderful job. Good job. Do we have any questions from the audience?
I have a small question. You've been working with our tools from our lab. I was curious how you found using like ComPreps and Fusion, the usability of it.
So we mostly just sent out our samples and received the annotations already. So I didn't heavily worked on them myself. But I think it was fascinating to see the integration of these tools that were created by different labs and see the power of the results that it can create.
Yeah, thank you so much. It's all about collaboration. That's why we're all here. So,
any other questions from our collaborators? Anyone else have a question they want to ask? All right, then I will ask, what was your favorite, like either research aspect or like result that you got from this project?
Yeah, I think this project exposed me to many various softwares and just having the skill of learning how to familiarize myself with those softwares and learning how to export one file into another and combining all of them was a very valuable skill for me to take with me.
Okay, another round of applause for her. Lovely job.